Hi biologists! Let's start by looking at the learning objectives for this lesson. Take note that this material is for the higher level biology only. At the end of this section and following the biology syllabus, you should be able to use the active site theory to explain enzyme function. What does this actually mean? What are we trying to understand? It is straightforward enough. You have to be able to use the active site theory to explain how enzymes work. In other words, explain the mechanism of enzyme action. Let's sort higher level enzymes by first looking at the active site theory. The substrate is the substance upon which an enzyme reacts. SUB in substrate, SUB in substance, represented here in the pink. The product is the substance the enzyme forms. In other words, the substance that's produced. Again, product and produced. The active site is that part of the enzyme that joins or matches the substrate, seen here. You might notice, to keep life easy, I have made the shape of my active site like a letter E on its back. Now, the original active site theory stated that the active site had a rigid shape, an inflexible shape. It stated that the enzyme had a rigid shape also. The enzymes functioned according to a lock and key model. The substrate fits into the active site like a key fits into a lock, and they had to be an exact match for the reaction to be carried out. However, it was discovered that the active site is a flexible depression on the enzyme surface. In other words, it's a hollow on the surface of an enzyme, and there's a certain flexibility attached to it. It is not a rigid shape. It's actually created because the enzyme is a globular protein with a three-dimensional or a 3D shape. And this 3D folding of the globular protein gives a three-dimensional hollow, which is rather flexible on the surface of the enzyme. It was also discovered that most enzymes are made of more than one globular protein that are called domains. And in actual fact, these variable domains, the globular proteins, can vary, so they join together to form the enzyme overall structure. Now we will use the active site theory to explain how enzymes work, or to explain the induced fit model. In this theory, the substrate causes the active site to change shape slightly. There's still a good match, but the shape of the active site is now flexible. It's maybe not 100% a carbon copy of the substrate, but it's a flexible depression, as we have said, and it can change shape slightly according to this theory. This results in a closer fit because the substrate and the enzyme snuggle into one another and fit more closely. The induced fit stresses the substrate, increasing the possibility of a chemical reaction. This theory is called the Induced Fit Model, and this is now the accepted theory for how enzymes work. Let's have a look at this a little bit more closely. The enzyme and the substrate match. The active site of the enzyme is complementary to the shape of the substrate. If that is the case, the substrate will enter the active site. The active site will change shape slightly, forming a snug fit around the substrate. The substrate and enzyme together form what we call the enzyme-substrate complex. This snugger fit stresses the substrate, actually puts the chemicals into a better position for chemical reactions, and thus the substrate will react to produce the product. This is rather like getting a bear hug from somebody. They hug you so tightly that your ribs feel they're fit to crack. The substrate becomes the products, leaves the enzyme. The enzyme is left unchanged. It is exactly the same as it was before. Therefore, it can be reused or recycled. This mechanism of action is called the Induced Fit Model. So what is the mechanism? Well, the active site shape matches the substrate, so the active site is still vitally important here. The substrate enters the active site. The active site is induced, or it changes shape to fit the substrate, and an enzyme substrate complex is formed. Now the enzyme substrate complex is the group hug. The binding stresses the substrate and changes it into the products. You feel as if your ribs are fit to crack. The products are released. The active site shape returns to normal, which means that the enzyme is unchanged and can be reused. They are very fond of the mechanism of enzyme action in exams, so make sure that you know these steps. Some added detail on enzyme action. Well, as we've said already, the substrate causes or induces the 3D active site to change shape. The active site fits more precisely around the substrate. The induced fit mechanism of enzyme action happens very quickly. Also, as the enzyme is unchanged, a small amount of enzyme can process a large amount of substrate in a short space of time. So there you have it. Now that we've reached the end of our lesson, have we achieved our objective? Can you use the active site theory to explain enzyme function?